Okay, let's start today webinar. I start now recording this part of the webinar. Then I will stop recording for two presentation, and then the last part of the webinar will be recorded. Well, good afternoon to everybody. I'm glad to see people that were following us for the iPlanta webinar program. This is not the iPlanta program. iPlanta is finished. This is a new project that I'm coordinating. It's an Italian network supported by the Italian Minister of University and Research, and it's specifically addressed to small RNA and peptides for controlling diseases and development in horticultural plants. The project has the aim to develop knowledge, materials, and interdisciplinary strategy to use small biomolecular, in particular peptides and small RNA, for contrasting pathogens growth and regulating plant development in important horticultural crops. And we focus on three crops that are important for our country, but not only, such as tomato, grape, and strawberry. And we see this as we try to explore this new molecule as potential sustainable alternatives to agrochemicals application. So this is the general aim of the project. The project is more or less in the middle, is a three years long project. We have 1 year and a half, and there are 5 partners: University of Ancona, Marche Politecniche, Verona, Milano, Padova, and CNR in Florence. And we decided now, because we have clearly also in this project the program of dissemination, and so this first webinar that we decided to organize is having the aim to start the dissemination of the activity that we are planning, we are including in the project, but at the same time it's also to create some further integration, some further connection with other researchers that are more or less linked to the research activity included in the project, that not in particular with some partners involved in this project. And I sent the invitation of this for these webinars also to the mailing list of iPlanta, because in some ways this Italian network is partially originated also by the Italian network of people that was joining iPlanta. And also for cost, for the cost organization, this type of activity are considered appropriate of high interest, because it's a kind of follow-up, can be in some way considered also as a follow-up of the previous project, and as an idea that we try to keep the network and maybe find out a new possibility of interaction, integration for making this network more sustainable, and keep collaborating and working on this topic that has been identified of very high interest. Well, I stop here, and this webinar today is organized in collaboration with, say, different partners, in particular with the partners in Verona University, so Barbara Molesini and Professor Tiziana Pandolfini, and so I leave Barbara to introduce the topic of today's webinar, and also to manage the webinar. Again, I keep recording until the end of until the beginning of the first speakers, and I will stop recording and I will start again for the last presentation. Thank you everybody for participating, and let's stay connected. Maybe we will be able to organize other webinars in the future. I will keep you informed. Thank you again for your participation. Okay, Barbara,
thank you.
it's your floor.
Thank you, Professor Mezzetti, for your introduction. And now we can move to the webinar, which will highlight the use of small molecules such as peptides, microproteins, and microRNA encoding peptides as potential strategies for improving traits of agronomic interest in a sustainable vision of agriculture. The webinar will consist of 5 presentations. If you have any questions during the presentations, please type them into the chat box. And I'll bring them up during the presentation, and we will have time for questions at the end of each seminar. And for the speaker, you have 20 minutes, and I will alert you when there's 1 minute left. And for the participants, please remember to switch off your microphone. Thank you. So it's a great pleasure for me to introduce Professor Stefan Wenkel from University of Copenhagen. Few words on his research activity in the context of the webinar's topic. The lab of Professor Wenkel in the past 10 years has identified and characterized microproteins, which act as post-translational regulators of the activity of target proteins through the formation of protein complexes. Thus, by altering the expression and the activity of these microproteins, it's possible to control specific traits such as flowering. The title of his presentation is Microproteins, Small Proteins with Big Impact. So, Professor Wenkel, if you're ready, you could start. Thank you.
Okay, can you hear me now?
Thanks to her, okay, thank you. And thanks to her, I experienced in the study of the molecular mechanisms underlying flower development and embryo sac cell differentiation, fruit development and ripening. In the last years, she's using peptide aptamers, which are short synthetic peptides able to specifically bind the target proteins, applied to plant biotechnology for controlling desired biological processes, such as, for example, also plant pathogen control. The presentation is entitled The Yeast to Hybrid Methodology, a microorganism-based strategy for drug discovery. Please, Simona.
Okay, let me know if I talk too low. So thanks Tiziana and Bruno and Barbara to give us the possibility to show what we are doing here in Milano. And I must say that drug discovery, it's a quite interesting topic. Actually, in 2015, the Nobel Prize given to William Campbell, Satoshi Moore, and UU2 indicated that there is the need for our society to find and to look for molecules able to counteract diseases for human beings. But this necessary is not only to counteract diseases, it's also interesting to counteract plant diseases in field. These Nobel Prizes were given to persons that actually look in a bit challenging way to solution against filariasis or malaria. Let's say that their research started in a way that was very similar to what Fleming did when he discovered the penicillin. I mean, he observed that a contaminated plate, like the one, like the one that is here, was blocked, the bacterial growth was blocked by something produced by penicillium, by a yeast, a contamination, by a fungus. And indeed, penicillin is still a widely used antibiotic. But nowadays, drug discovery wants to be more structured in a way that researchers wanted to search for molecules able to target specific protein-specific enzymes in order to optimize the efforts and to reduce the challenge of the search. And basically, this fishman represents what drug discovery is nowadays. There is a bait of interest, the protein of interest, and an ocean of fishes that are the molecules that might interact with the target and might prevent its action, whether that's the desired target. So the identification of what the fishermen use to look for fishes is very important. And indications are given, of course, by basic research, by genetic studies, biochemical studies, physiological studies, and so on. And as more as a target is appropriate, as more the challenge to find a magic bullet is high and to land on very interesting molecules that can go out from the lab finally and join the market. That's the possibility. Applications are, of course, regarding in biomedicine, but also in veterinary, in feeding stock, but also in agriculture. And our group is looking for a system to screen very quickly and whether possible also in a quite cheap way, why not, a huge ocean of fishes to find the molecules that might interact with the desired target, with the bait. Of course, this lake is not crowded enough. What you need to look is to really millions of molecules. Only in that way, the screening will have a high possibility to find molecules that might change our life and might improve our life. So how to do that? Keeping in mind that we want also to address economical question. I mean, we want or we would like to have economical screening. So in our group, we are quite good actually with yeast to hybrid. It's a technique that we are using since now 20 years, even more. And our idea was to use yeast, that is a Saccharomyces cerevisiae, a living organism, for screening for molecule, for small peptide that might interact with desired target. So we adapted an yeast to hybrid approach to look for small peptides. I must say that Saccharomyces is a microorganism widely employed in our life. It's used for bread for preparation, for wine preparation, but it's also a very interesting lab microorganism. And everything started now 5 years ago, basically, when a local foundation from the city where I live and where I work gave us the money to prove that the idea that we had was successful. So, and this was absolutely in line with the view of the actual president of the European Community, Ursula von der Leyen. So the idea was, can we convince the sponsor that we are able to design a rapid, quick, and cheap pipeline to identify new drugs using yeast? And if so, which is the pathogen that we want to address? So when we submitted the project to the Caripol Foundation, and I must thank especially Paolo, the coordinator, we thought that Plasmopora viticola, an omicet that attack grape, could be an interesting target because several treatments are necessary to counteract its diffusion. And these treatments are, how to say, are economical relevant, but also are environmental relevant. So they leave traces. So that was our target, and the yeast to hybrid was our tool. How we arrived there? So for those that are not familiar with the yeast to hybrid, the yeast to hybrid is a methodology that allows to detect protein-protein interaction. And it's, how to say, just a first indication that the two protein interacts, of course. Then you will need more studies to prove that the interaction is occurring in vivo. The system that we use is based on a yeast transcription factor, the GAL4, which has all the transcription factor, is able to bind DNA. And the DNA binding occurs through the GAL4 binding domain, GAL4BD. And as several transcription factor, it's able to interact with the transcription in machinery. this occurs from the GAL4 activation domain. In the GAL4 transcription factor, these two domains are very close, but in the two hybrid system that we use, they are split in two different chimeric proteins. Fused to the GAL4-BD, there is the bait protein we wanted to study, so this is the bait. We'll fuse the GAL4 activation domain, and normally there is a library. And what we attached, we fused it in frame to the GAL4 activation domain was a library containing linear peptides of 8 amino acids. If the interaction between the bait and the peptide occurs, then the GAL4 activation domain and the GAL4 binding domain are close enough to activate the reporter gene, one or more reporter gene, which expression is driven by a promoter recognized by GAL4 transcription factor. Normally, these reporter genes are genes that allows yeast to grow on media that lacks relevant, important, essential amino acid, such as histidine-free. We also like very much as a reporter adenine. So this is a typical plate on the left that comes from a screening. You have one colony that survived, and this colony alone that you can see on the plate means that there is interaction between the bait protein and the product of the library. How is the library? Well, the library we couldn't attach to the GAL4 activation domain, that is a quite big domain of about 100 amino acids. We couldn't attach 8 amino acids. That will be completely included in the folding of the GAL4 activation domain. We fused it in frame first thyrodoxine from E. coli. Then we inserted in the thyrodoxine our 8 amino acid, here indicate with N and K. And we knew exactly where to insert them, so in a way that they could protrude on the tip of the thyrodoxine that acts like a finger-like structure, and on the tip of the finger there are the linear peptides, so our library, our fishes. So to make a long story short, as I said, we looked at something that could block Plasmopora viticola, and in particular, we were focused on the zoospore germination. During germination, this zoospore of Plasmopora viticola produced a cell wall, which is mainly formed by glucans, and we thought to use for our aims as target, as bait, to use enzyme involving the cell wall formation. The bait that we chose during this project was Plasmopora viticola cellulase synthase 3. Omics that are not true fungi, they have a cellulosic cell wall, and we screened in our library. We got about 5 peptides, but I'm going to show you the results of the best one that we got, that we call it the no PV1, so no Plasmopora viticola 1. And this peptide actually indeed works. So we treated the leaf disc, but we also treated plants and greenhouse with it, just solving in water the peptides, and we could get a protective effect on the leaves of grape. So we were quite satisfied because pipeline was satisfied, and we could give back to the sponsor a proof of concept quite important. So if we look at SEM, what happens is that the zoospore, as you can see here, where there is no PV1, do not germinate at all, or if they do, the appressoria move not towards the stomata as normally does, but in a very disordered way. So we had the bullet, but more importantly, we find or we design a way to identify new peptides, whatever we wanted to do against what. We also realize, however, that linear peptides are not easy to deal with. And to favor the cell penetration, to favor the affinity, and to favor also the stability of these molecules, better to look for cyclic peptides. So we asked ourselves whether is it possible to generate yeast libraries to screen cyclic peptides. And I must say that a big thank goes to 2 persons. To Bruno Mezzetti, that coordinates a national grant where we are involved, and to Paolo Pesaresi, that coordinates FET-open, that we are also involved as a unit, and that gave us the possibility to generate a huge library. So right now we are able to screen almost 10 to 7 independent circular peptides with one screening. So these two projects gave us the possibility to extend the pipeline that we had and to improve greatly. So hereby the person that are doing actually the work for these two projects, mainly Stefano Rosa, my PhD student, Andrea Tagliani, a postdoc fellow, and Chiara Bertaso, a diploma student, that generated the Cyclic, that is a new library of cyclic peptides, and the cyclization occurs thanking intane. Intane is a self-protein that is designed by evolution to do something just to maintain itself, and it's simply spliced out by the protein where it's transcribed with. During the splicing of intane, it's possible to get the circularization of what it's included if you change in a proper way the amino acid that franks the intane C and the intane N. This Western blot that we have run confirms that indeed in our yeast colonies, cyclization occurs when we insert between intane C and intane N, again, our cyclic peptides of 8 amino acids. So that's how the library looks like. In end, we have the Gal4 ED, intane N, it comes cut away from the constant, from the final protein, and what it's retained, fused in frame to the Gal4 activation domain, is intane C, that is quite small, not many amino acids, plus the library, that is circularized. So with this library, we went back to our main problem, that the OMI set, and we screening for more targets. The library that we generated is actually very good. So we had most of the clones, 86.5% that are in frame, that made us very happy. And more than 50% of the insert are able to circularize. So we were quite happy. We could not avoid to completely reduce the codon stop insertion. So that's the only thing that we regret about our library. Okay, so thanks to the project that I mentioned you, we are able now to screen for peptides able to interfere with the OMI set. We decided to focus on this pathogen, and we made a jump. So we decided not to screen again for Plasmopora viticola, but rather to look for molecules able to interfere with the worldwide problem that is a Phytophthora infestans. Again, we thought that to look at the zoospore germination and the cell wall formation might have been promising since we had good results in the past. And we then cloned and screened cellulose synthase A3. Actually, we are screening for all the cellulose synthase of Phytophthora infestans and also for the chitin synthesis as well and argaglucanase. But to make a long story short, we have done more than 20 screening. I will present the best peptide that we have so far isolated. So using cellulose synthase A3 as bait, we have been able to isolate something like 25 independent peptides, most of them appearing several times in the screening. And CP20 is indeed the most promising. Here it is. So CP20 works with, this is in the graph, there is its EC50 line works at minimum concentration of about 15 micromolar. So works with a very low concentration, blocking Phytophthora infestans zoospore and mycelium both growths. So this is a treatment with sporangia. What you can see up here are the not treated sporangia, whilst these are the few sporangia that after 3 days can try to germinate. But indeed, when we run a trypan blue staining, these sporangia are dead. Again, the concentration that we used for the experiment was 20 micromolar. So Since cellulose synthase is quite conserved among omisate, we tested whether CP20 can block also closely related pathogen to Phytophthora infestans. And again, it works with Phytophthora capsici and with the Plasmopora viticola. So again, this confirmed that what we have generated in the lab is really a good pipeline to find drug and to be used not only again for agriculture, but also for other aims. Of course, one might say that, okay, we were lucky. We had indeed found simply antimicrobial peptides that can interact with the membrane, with the plasma membrane of the omisate. And I want to convince you that indeed we are sure that the block occurs from protein-protein interaction. And we went back a bit on the library. We wanted in this case to make our life easier, and we wanted to fill this last tile in the puzzle. So what we have done is to study all the peptides that we isolated that indeed, are able to interact with the Gal4-BD. Because anytime that we run a screening, of course, we have peptides against our bait, but also, of course, against the Gal4-BD that is 107 amino acid long. So it's a small itself bait. These are a list of peptides. We all assayed against Gal4. And we can say that we have a good indication, biochemical indication, that at least some of them interact directly with the Gal4 binding domain. We checked the literature. This was the work of Stefano and Andrea. It took them quite a while to find the phenotypes in wild yeast strain. And what they have found that lithium can induce galactose, can induce toxicity once Saccharomyces cerevisiae grows on galactose and that's it, without glucose. What you can see, look at A2, these are spots of yeast grown on lithium chloride at high concentration, but also in presence of galactose. And as you can see, the peptide A2 can overcome the toxicity induced by lithium. So that's a proof. But we were not happy enough. So what we have done, it was to do a Western for the YFP. And the YFP is detected once that the yeast colonies grow on galactose, because YFP expression is driven by a promoter recognized by Gal4. So only if Gal4 is there, YFP is transcribed and translated. But if there is a 2, you cannot see anymore the presence of the reporter, meaning that the binding of Gal4 is prevented and YFP cannot be activated anymore. So we are now, right now, doing cheap experiment to confirming how the pattern of the Gal4 binding is occurring. But again, this indicates us that what we have designed is a tool that allows us to have antimicrobial peptide, yes, but the toxicity of them for the pathogen occurs because they can specifically binding the target without no off-targets effect. So I wanted to thank the group that is here in Milano. We collaborate for the cell wall part with the KTH, the group of Vincent Bulone. And a special thanks to the guys that are really doing the work, not with me anymore in the lab, I must say, I'm behind the desk, unfortunately, Stefano, Andrea, and Chiara. And a special thanks to the person that believed in us, that is mainly Paolo, because he was the only one to believe that this could have been challenging 5 years ago, and to Bruno after that. So thanks a lot, and that's it.
Thank you, Simona, for your presentation. Are there any questions? I don't know if I've lost some question in the chat, but
I cannot have
yeah, I think that at the moment we haven't any question.
Oh, I think one question since
sorry,
the old one.
Also,
Since the mini prep is the same in the treated plants, but overextracted, it should not be a problem.
Is
Calida Mare. Well, okay, I don't know. Maybe Khalid can ask.
I think that we can, at the moment, move to the next speaker.
Okay.
If you have any question, we have time at the end, I think. So the next speaker is Professor Tiziana Pandolfini from University of Verona. Her research activity is mainly aimed at studying the first phase of fruit development and in particular at identifying key genes involved in the transition from flower to fruit. Among these, tomato cystine-knot mini proteins emerged as versatile tools for different biotechnological applications from plant development to pharmacology. And the title of her presentation is Cystine-knot Peptides and Tomato Fruit Development. Please, Tiziana, you could start.
Okay, just a moment. Can you see the...
Yes.
Okay, can you see the presentation? So good afternoon to all. And my speech will be about cystine-knot peptides in tomato. So we have studied for a long time the first phases of fleshy fruit development, and in particular parthenocarpic plants. And during the study of transgenic parthenocarpic plants, we came across of these particular peptides, and we got interested in it. Cystine-knot peptides are small cystine-rich proteins that are present in several eukaryotes organisms and are characterized by the presence of 6 cysteine, as you can see here, that form 3 disulfide bonds that are arranged in a knot. This particularly three-dimensional structure, which is very compact, confer to this peptide some very interesting characteristics. For instance, they are resistant to extreme pH or high temperature, and also proteolytic attack. So this has attracted the interest in very field for biotechnological application, in pharmaceutical biotechnological application, and also agronomical application. So in plant, cystine-knot peptides are well represented in different family and can be linear or cyclic. And they range from, they have different activity, biological activity, and of course are grouped based on this activity and also on the sequence similarity. For instance, there are some alpha amylase inhibitor, serine peptidase inhibitor, some toxins, and some insecticidal insecticidal peptides. And we have focused on a group of these cystine-knot peptide, which are called metallocarboxypeptidase inhibitor, and this name came from one of the first cystine-knot peptide that was detected in a potato, and and has a very high inhibitory effect of metallocarboxypeptidase. And another interesting feature of this group of cystine-knot is that they are restricted to species belonging to Solanaceae. So here we see the phylogenetic tree, and there are several members in potato, in tomato, in tobacco, and maybe there are also other which are not yet identified in other Solanaceae species. But we don't find this type of cystine-knot peptide in other plant family. So we focus on tomato cystine-knot peptides, and at the moment there are 13 members of this family that has been identified, and more or less all these members have relatives in the wild-type tomato. So in Solanum pimpinellifolium, that you see here, which is the most probably the progenitor of the domesticated tomato, and also in Solanum pennelli, which is another wild-type tomato. So it seems that during domestication, these protein have been conserved. There is an exception, and in fact, in Solanum pennelli, one of these Solanum lycopersicum, one of the most studied Solanum lycopersico cystine-knot peptide are not present, but is the only one exception. So we have screened the majority of these cystine-knot peptide, analyzing their pattern of expression, and most of them are expressed in all type of tissues, starting from the roots, the leaves, and also flower buds, fruit. But there are two tomato cystine-knot peptide that have a very peculiar expression pattern, and we have named TCMP-1 and TCMP-2, which are highly expressed in the reproductive organs. And for instance, TCMP-2 is specifically expressed in fruit, as you can see in this picture, are highly expressed in the seeds and also in the vasculature of the fruit, but are not expressed in normal condition in leaves or in other part of the plants. And in fact, the promoter of this gene, which is also called 2A11, has been used several times to express some genes specifically in the fruit. So this 2-TCMP has really a very characteristic expression pattern during different phases of flower and fruit development. TCMP-1 is highly expressed before anthesis. This is a Northern blot analysis, and then after fertilization, there is a drop, its expression drop, and then it's expressed a little bit also during the early stage of fruit growth. While TCMP-2, as we have said before, his expression increase during the different phases of fruit growth until fruit maturation. So this very peculiar expression pattern reminds to some coordination between the expression of these two genes. And indeed, we perform a chromatin immunoprecipitation, demonstrated that both genes are contains in their promoter several response elements for a common transacting factor, which is the inner no outer INO transcription factor, which is a member of the YABI family of transcription factor, and is involved in the regulation of fruit development and is expressed in the ovule, exactly in the outer integument in the ovule. So this kind of, we have then the idea that the expression of these two genes were in some way coordinated. And so our idea was to unbalance both the level of this protein and also their ratio. And to do this, we decided to build a construct where the coding region of the TCMP-1 was driven by the promoter of TCMP-2. This construct was used to transform tomato, and then we observe really changes in the expression level of both genes. So indeed, we had a alteration of their expression ratio. As expected, TCMP-1 was expressed in the fruit, where normal in the mature fruit, and in the green fruit, which is normal, not expressed, so in the later phases of fruit development, while the expression of TCMP-2 remain unaltered. But the measure differences, we observed the measure differences in the first phases of the fruit development. So in the phases from flower buds to flower anthesis. And here we have a reverse of the expression. In fact, TCMP-1, which is normally in the wild type, highly expressed before anthesis, so before fertilization, in the transgenic lines was reduced, whereas TCMP-2, that is normally expressed at very, very low level in these phases, was induced. So what was the effect of this alteration? Then we look at the phenotype of our transgenic plants and at different stages of reproductive development, and we did not observe particularly changes in the morphology of the fruit, in the weight of the fruit, or in the seed number, a part of a little delay in one of the two lines in the time from the fertilization to the breaker stage. But what we observed was a differences in fruit set, so in fruit initiation. In fact, our transgenic lines produce a number of fruit, an elevated number, a greater number of fruit than wild type plants in the first stages of the reproductive development, and so we have an earliness in the fruit setting. And another striking difference was detected in the flowering phenotype. In fact, flowering phenotype in tomato is quite particular because they are sympodial. There is a sympodial habit. So we have the first inflorescence about after 9-10 leaves. Then the first after the production of the first inflorescence, we have the termination of the first axis. Then we have the sympodial growth, and in this sympodial growth we have a decreasing number of leaves, and after the complete termination of the sympodial units. Whereas in our transgenic plants, we observe that an accelerated termination. So the sympodial units arrive to produce flowers at a very early stage. Indeed, in tomato, there is flowering is a phenomenon that is regulated by a competition of key factors that stimulates vegetative growth, so anti-florism, I would say, and key factor that stimulate flowering, for instance, the orthologo of the FT gene, which is the SFT. And indeed, in these plants, we observed that SFT was the expression of SFT was induced, while the expression of the SP, self-pruning, which is a gene that that maintain the vegetative growth of the plants, so the production of leaves, do not change, and also constance was not altered. The expression of constance was not altered. So our plants has having altering the expression level of these two genes in the first phases before fertilization, we obtain an increase in fruit set and also an accelerated termination of the sympodial units, while no differences was observed in the formation of the first inflorescence. So to have an idea of the mode of action of TCMP in fruit set and flowering, we perform a histlohybrid screen. So we use as bite the cystine-knot domain, here highlighted in orange, and we use for the screening a library of a cDNA library of different phases of the different reproductive phases of tomato. And what we found that one of the interactor of TCMP was a B-box protein, the tomato B-box protein number 16, and we confirm by again this interaction by histlohybrid, and also we prove that TCMP interact with a close homologue of the BBX16, which is BBX17. And using quantitative bimolecular functional complementation, we prove that the interaction between the TCMP domain and the B-box protein was due to the presence of this domain in the to the presence of B-box domain in the B-box 16 protein. So as maybe you know, BBX protein are a large family of protein that are involved in many developmental processes and are different. They are different group of BBX that are grouped based on the presence of different domain. Some of these protein contain two B-box domain, B1 and B2. Other contain also a CCT domain, which is probably responsible for their activity as transcription factors, and other contain a single B-box domain. So our two tomato BBX are of this last type, are small. They contain only one BBX domain, and they are very similar to Arabidopsis microprotein MIP1A and MIP1B. And so we performed also an analysis and twist hybrid analysis to prove the interaction of the TCMP also with the Arabidopsis MIP1B. So Arabidopsis, in Arabidopsis, microprotein P1A and B are are involved in a complex with the constants and are also responsible for the recruitment of topless, which has an inhibitory effect on the expression of the florigen, FT, and so the overexpression of this MIUR in Arabidopsis results in late flowering. And when we express our TCMP-2 in Arabidopsis thaliana, we observed indeed an early flowering, the opposite effect. So we see that we have a decreased number of rosette leaves at bolting, and also an increase in the expression of the florigen gene. And so one of the hypotheses that this effect of the overexpression of TCMP2 in Arabidopsis thaliana could be due to could be linked to its interaction with Arabidopsis MIUR1 A and B. So going on, we were interested to analyze the role of BBX16 and BBX17 in tomato, but first of all, we try to express these two tomato protein in A. thaliana to find out if they in some way phenocopy what happened when a TMIP1 A and B is overexpressed in Arabidopsis thaliana. And there are preliminary results, but we have observed that there is indeed a slight late flowering, which is more accentuated in when in the plant that overexpress BBX17. And of course, we have to go on with the molecular analysis to find out whether this tomato BBX protein can in some way phenocopy the effect of MIP. And of course, in progress, there are also the overexpression of this tomato BBX in Arabidopsis thaliana, in a MIP1 A and B knockout mutant of Arabidopsis thaliana. Then, of course, at this point, we are also interested in what happened in tomato, because, you know, in flowering tomato and also fruit set is very different from Arabidopsis thaliana. So we don't know, for instance, which is the contribution of constants, the precise role of constants in this in the flowering phenotype. And also tomato is a day neutral plant, so they don't respond to the regimen, to the light regimen, and and we don't know also if there are some complexes like that described in Arabidopsis that operate in tomato. So first of all, we know that BBX16 is expressed in the leaves as a TMIP1 in Arabidopsis, but we have also seen that these proteins are expressed in all the stages of the flower and fruit development, whereas the expression pattern of BBX17 is really very peculiar, because it seems that it's expressed at very low level in the vegetative organs of tomato, but is expressed at high level just before anthesi, and then dropped after anthesi. And this expression pattern is quite common in protein that act as repressor of fruit set. So now we have expressed the we have overexpressed the two protein in tomato, and we have also we are going to screen and to analyze the phenotype of these plants, and we have also silenced the BBX17. So to conclude, what are the potential applications of this cystine-knot protein in agriculture? We have seen that the first member of this family, the potato metallocarboxypeptidase inhibitor, has a strong effect against carboxypeptidase. So the first thing that you think about its role, it's a defense against the pathogen, and indeed there are examples of overexpression of this potato cystine-knot in, for instance, in rice, and they have observed that this expression increase the resistance both against fungal pathogens and also pests. And we don't know if others member of this family possess this type of activity. We have never tried to see if our plants with our transgenic plants have different behavior against pathogens, but it would be a possibility. The other possible application is to tuning the expression of this TCMP, cystine-knot peptide, using appropriate promoter in order to control flowering. So to have, for instance, a different timing of flowering in different varieties, in different type of tomato plants, or increase the fruit set. Another possibility is to use the exploit the interaction of this cystine-knot peptide with other proteins, so to screen the possible interaction of TCMP with other protein that are, for instance, topless or constants that are involved in protein complexes that with regulatory effect on fruit development. And okay, the last application, it's possible to, it would be possible to use this peptide as exogenous bioactive molecules. We don't know. We have few information, I would say no information about the possibility to use this TCMP, and as Combier have highlighted, there are several problems with the ectopic use of peptides because the way of delivery, but it seems quite easy to deliver, but also the cost of production and the dimensional structure. So the effect of the cystine-knot peptide is connected to their characteristic three-dimensional structure, and so they don't have the same activity as linear peptide. Okay, and then there are, I want to thank all the people that are involved in this, have been involved in this project, and also the support from the PRIN project, and in particular I would like to thank Barbara Molesini, Valentina Dusi, and Federica Pennesi, and all the other collaborators from the University of Verona and from other university and research center for agriculture.
Thank you, Tiziana, for your presentation. We have time for only one question at the moment, so take other questions for at the end. The question is: it looks that the TCMP-2, TCMP-1 promoters are regulated through epigenetics. Did you verify the respective DNA methylation of these sequences?
So it's a good question, and it's a possibility. At the moment, we haven't verified the state of DNA methylation of the promoter, but it's a good idea to do it.
Thank you, Tiziana. And I think that we must move to the last seminar from Professor Thomas Dresselhaus. Good afternoon. And we are recording the webinar, Professor Dresselhaus. Do you prefer that we stop the recording if you are presenting unpublished data or for any other reason?
So
we can stop if you prefer.
So I don't mind. So you probably don't send this somewhere accessible to everybody. So it's in your closed club, I hope, or?
Oh, it will, sorry, I'm Bruno. It will be published on the web page of the project, the link of the recorded webinar. So it will be public.
We can.
Then I really prefer to maybe stop here.
Okay.
Crossing or hybridization with other species. Yeah, by allowing also alien pollen grains to penetrate, for example. Okay.
Any further questions?
A general question about Ralph gene. We know that is a group of genes that are involved also. Peptides are involved also in disease resistance. So do you see some kind of connection related to this mechanism and disease resistance? So are you studying also this aspect?
Yeah, I did not get all of your questions. Sorry. So you
about disease resistance.
Yes.
The role of the genes and the peptides in the disease resistance.
I think that's an extremely interesting aspect because it's partly absolutely identical receptors that are used for reproduction, for immunity or immune signaling. And the interesting thing, for example, is of course one of the interesting candidates is Feronia, that plays a role in vegetative development, in root hairs, in immune signaling. We see it at penetration of papilla hairs. We also see it during pollen tube burst. And it's always the question, of course, how active is Feronia signaling? And so which other components are available? So Feronia, other receptors, of course, they also partly function as scaffolds to recruit other proteins that are present. So the question is partly the specificity is regulated because certain receptors, co-receptors, or peptide ligands are expressed, and therefore you can use the same molecules for completely different processes. And downstream signaling, so for example, for these receptor kinases seems to involve very often ROS, but then also ROS, it depends on the ROS levels that you generate in the end. If there are very high ROS levels, they are toxic. They are toxic for pathogens. If you have middle ROS levels, they could be used for long-range signaling. And so I think also this and the whole complexity that we see is probably to fine-tune, for example, the level of ROS, the level of downstream signaling processes that in the end could lead to inhibition of growth, acceleration of growth by changing actin cytoskeleton activity and so on. I don't know whether that answers your question, but I find this extremely fascinating. That and also my hypothesis is that we had immune signaling long before, and once plant reproduction in angiosperms was developed, they used signaling systems that were available, and that's why we see such a strong overlap between reproductive signaling and immune signaling. My hypothesis is immune signaling was first, and then after establishing new organs, new functions in flowering plants, they simply also used what was available.
Thank you.
No other question? I think that we have finished with this presentation, and I would like to thank again all the speakers for their very nice presentation and all the participants, and I'll give the floor to Bruno Mezzetti for the final.
No, thank you, Barbara. Well, thank you to you for managing this webinar, for organizing and managing this webinar, introducing the speakers. I want to thank all the speakers for really the height of presentation, high quality presentation. And well, it's the first time that we do this type of webinar for our Italian project. I hope you enjoy it. We have a very high number of people participating. Following, we reached more than 100 people. Sorry that we were not able to record, but I understand what are the needs of the different speakers. So for the people that want, we'll be able to find what we had recorded on the web page of the project. Well, if you like, we can take a virtual group photo. We are used to do that. So you can just turn on your camera. It's something, we need to do dissemination. So anything that help to demonstrate that we have been active, it's always helpful. So if everybody can turn on the camera, we can still few are missing from my side. Maybe also Livio can take some pictures, so we are sure that we get more complete one. Okay. Well, that's it. Thank you very much, everybody. Thank you again, the speaker, Barbara, and goodbye to everybody. And let's see if we can meet again next with another webinar related to other topics that are included in the project. Have a nice evening. Goodbye to everybody.
Goodbye.
Bye-bye.
Bye.
Bye.
Bye-bye.
Bye-bye.